Hello again, and welcome everyone to our sixth lecture for the summer school from the brain simulation section on TVB. And today, my colleague David and me, Luise, we will talk about image processing workflows. But why is processing such a thing? Or why do we dedicate one whole lecture to processing and quality control? To illustrate this, let's take a look on this study by Rotem and her colleagues. They provided the same fMRI dataset to 70 independent research teams and asked them to test 9 exactly the same hypotheses. And this figure visualizes what happened. Each branch represents one decision that is made during the processing or the analysis. So for example, which processing steps do we take? Which one do we neglect? Or how do we define the statistical thresholds? And with every additional decision that we take, those paths are diverging. So by the end, the teams had arrived at substantially different results. So we see that we need to take every decision quite harshly, and that every decision furthermore induce another degree of freedom. And those decisions can, at the end, ultimately influence also our scientific conclusions that we draw. This said, I hope this underlies why we need to be extremely careful also during processing, and then that we need to include the decisions that we took during our processing steps afterwards in the interpretation of our analysis as well. Here is our agenda for the day. So first, as processing is quite a complex field, we will talk a bit about the basics and also induce some important vocabulary to understand the pipelines afterwards. And then we will talk about 2 different post-processing pipelines. More specifically, I will talk about the structural MRI processing pipeline, and David will talk about the functional MRI processing pipeline. This hopefully will be around an hour, and then we will have a small break, and afterwards we will talk about, or we will dig deep more into quality control and have a practical hands-on session. So let's start. This video you probably already have seen quite often. This shows how we construct a TVB model. So we need the brain anatomy, we need the tractography of the brain, and then we also have to know how our brain is parcellated so that we can group different voxels into one region or one node, so that at the end we can construct our network. This network, so to say, is built from the structural connectivity, which incorporates the information of the tractogram of the brain and the brain anatomy and its parcellation. We have also known so far that we also need a functional connectivity, which consists of or takes the information of the brain activity. More precisely, for example, this region here would show the correlation between brain region I and brain region J. And all of this information we gain out of MRI images, more precisely diffusion, structural, and functional MRI. And today we will talk more about the construction of, or how we compute the functional connectivity. We decided to give you a more in-depth and practical lecture today, and therefore we had to prioritize a bit on what we talk and what we have to neglect. However, Micha already presented to you this paper or talked about how important containerized workflows are, and here you can read about them. And he constructed here the full pipeline from all different MR modalities to the last output for TVB. Okay, so all of those modalities that we just looked at, just to have a small summary of them, we have the structural MRI. here we need the anatomy of the brain and the brain parcellation. We have the diffusion MRI, which shows us the connections between the different brain regions. Interestingly, this is a really nice image, and in 2014 it won the Image Award prize, and it was made from Zeynep. I just want to give her some benefits here as well. And this one is functional MRI. Here we gain the brain activity, so we see which specific parts of the brain are active in the moment. And all of those different MR modalities are now underlying the same physical principle. To understand this, we have to look at the MR scanner itself. Here you see the scanner and also the huge coils in the middle of them. These super large coils are building a superconducting magnet. The fundamental principle behind it is electromagnetism, or more precisely, Ampere's law. So if electrons are flowing through a conductor, they are creating a magnetic field that is then wrapping around this wire. I'm not sure if you had this at high school, but we learned it with the right hand rule, so that if you take your hand in the direction of the thumb is where the electrons are flowing, and then you have the magnetic field going in the direction of your fingers. And this field now goes through the full scanner, and we call it B0 field. We also need an additional second magnetic field, which is built by those radio frequency coils. There is an alternating electric current going through them, so we generate pulses of a smaller magnetic field. This said, we can now zoom into our patient, or more precisely, we need to look at the water molecules inside of our body. Water molecules consist of one oxygen atom and two hydrogen atoms, and hydrogen has proton in its nuclei. And every protons have a nuclear spin, and therefore as well nuclear magnetic momentum. Here we represent it as an error. And in the absence of a magnetic field, these spins are randomly distributed. If we now apply our B0 field, they all align. Now, remember we had two magnetic fields, right? So we apply the regular, the B1 field, or the radio frequency pulse, and those spins realign and then jump back to its equilibrium. This time here, called T1, the recovery time, is measuring the time between the radio frequency pulse and the measurement of the signal for the spins to realign to their equilibrium. We can also measure a second time, which is called T2. This signal we measure, or shows how long the spins take to react to the radio frequency pulse. And this we can model, or we can control by changing the repetition time. This is the time or the frequency of the B1 pulses. This said, we now already have the full physical background and the vocabularies that we need for processing. Just as a summary, we have two magnetic fields, B0 and B1. B0 is the huge one. This is important. We have the relaxation time, and we can measure two different signals, the T1 and the T2 time as signal. So T1 is the recovery, and T2 is the signal decay. And we can control the repetition time, meaning the frequency between radio frequency pulses. And this is basically it. This is everything that we need to know to understand how we can build different MR modalities. So if we look at structural MRI, here we take into account that different tissue types have different relaxation times. And therefore, for example, here, this is a T1 weighted image. We see white matter as light, quite light, and gray matter here as a bit more darker, and CSF as almost super dark. We have diffusion MRI, where we take into account that water is moving throughout the brain. So water is moving in the direction of our fiber tracts, and this movement we can track afterwards, and therefore we can reconstruct the fibers inside of our brain. And then we have also functional MRI, how I already mentioned before. Here we measure more the oxygen changes inside of the brain, which means how much usage of oxygen in specific brain parts we have. So we take this as a proxy for brain activity. What I mean by that is if we have a high neural activity, we also have an increased oxygen consumption, therefore even larger blood flow to these specific areas in the brain, and then also a relative increase in oxygenated hemoglobin, so a higher Bolt signal. Until now, do you have any questions considering the physics behind MR imaging, or can we continue? Okay, nice. Then I would say we just continue with David's part.
Hi, everyone. And thank you, Luise, for explaining the physics background behind MRI signal acquisition to us. And in my part, I'm going to try and bridge the gap between this theoretical background and then what you actually experience when you try to work with MRI images on your computer. So the space inside the scanner can be divided into voxels, which is basically the 3D version of pixels. So these little cubes you can see in the right brain. And the way we acquire MRI signal is that for each of these voxels inside the scanner, we have one signal intensity value. The resulting image is therefore either a 3D matrix for structural images or a 4D matrix for functional images, where we get one 3D matrix for each time point in the time series. Could I get the next slide? Yes, but the image matrix itself does not contain any information on how the image was acquired or any other important data. And therefore, usually when we store an image in a file, it consists of both an image matrix and metadata. Next slide, please. And there are two basic image file types, MRI image file types. The first one is DICOM, and that one was developed for clinical use and is optimized for the way clinicians inspect MRI scans, which is usually visually and only one scan at a time. And DICOM, therefore, is a bit cumbersome, but it contains a lot of details about image acquisition and also about the patients, which makes it useful for clinicians. However, it's not very suitable for analysis, and therefore, people came up with the NIfTY format, which is optimized for research, where researchers usually analyze data computationally and with many images at the same time. And for that, NIfTY omits quite a lot of metadata. It only stores image resolution and orientation. And this makes it small and efficient and enables this sort of like large-scale computational analysis that we usually do. And lastly, sometimes it is preferable to analyze the cortex not as a 3D volume, but rather as a 2D surface reconstructed from the 3D image. And I believe Luise will talk a bit more about this later. But for this purpose, people came up with SIFTY format, which is an extension of NIfTY, which allows us to store this 2D cortical data alongside the volumetric data about subcortical structures. Next slide, please. So the typical Nifty file will consist of an image matrix, then the header with metadata, and then a affine array. Next slide. And to understand what the affine array is for, we first need to talk about different reference spaces. In an MRI image, each voxel can be identified by a set of 3 coordinates. However, these 3 coordinates only tell us where within the 3D image matrix the voxel is located. That is, where its position in the voxel space. However, these coordinates tell us nothing about the voxel's position within the scanner or relative to the subject. And these other reference spaces are important since different modalities can capture different parts of the brain or have different orientation, as you can see in the picture on the right, where the T1 weighted image was taken with one field of view and one orientation, whereas the bowed image was taken with a different field of view and different orientation. To be able to identify the actual location in the brain to which a voxel refers, therefore, we need to project those voxels to some common frame of reference, for example, the subject space. And the affine array is basically a matrix that describes a linear transformation that does this projection from voxel space to subject space. There is one more space that we should care about, and that is some sort of normalized space, because when we do group analysis, different participants have brains of different sizes and shapes. So to compare them, we also need to project them to a common reference space. And the most common of these normalized spaces is the Montreal Neurosciences MNI space, which was basically produced by averaging a large amount of individual brain images. Next slide, please. Okay, so I hope now we have a good image of what is contained in a single image file. However, usually we do not deal with single files because we have multiple subjects, multiple sessions, runs, experimental tasks, modalities, and so on. Therefore, we have quite a large amount of files, and it would be useful to have some standardized way to organize them. And that is exactly what the brain imaging data structure is for. It provides this standardized way of how to organize your files, and this allows for easy comparison between datasets and also for automatization because computational tools and pipelines require predictable data structure. For this reason, it is usually the first step in processing to bitsify your dataset. And this can be done using some automatic tools, although it is important to remember that these don't work perfectly, especially when the original data in DICOM is not in some predictable structure already. Next slide, please. So the file names in bits have a specific structure, and the goal of this is to come up with a unique name for each file in a systematic way. So each file consists of a number of key value pairs, which describe the subject, session, experimental task, and other descriptors that distinguish this file from other image files. Each file name then ends with a suffix, which describes the imaging modality. And for each image file, there is an accompanying JSON sidecar file, which contains metadata about the image. Okay, does anyone have any questions about this part that I just went through? Great, then I hand over to Luise again.
Thank you, David. Well, now we know all the basics, so we can hopefully start to look into the processing pipelines itself. So now we look somehow how to get from these images, like the raw MR images, to these beautiful pictures. You already have seen this overview, and I will start now with the structural MRI imaging. So, how do we get from structural MRI images to the surface, or more precisely, the parcellation of our brain? So let's start with the first step, the bias field correction. Here you see the bias field inside of the scanner. And you see that this field is inhomogeneous. So this means that also the brain inside of our scanner would be inside of, like, different parts of this brain have a different magnetic field strength. This would result in an MR image with some weird intensity fluctuations or variations. Well, now we can say maybe that's not important. Not really at all, because most of the following algorithms that I will present to you are based on different intensity values for different tissue types. And therefore, we have to model the bias field as a smooth spatial variation in signal intensity and can then distract it from the image. So mathematically speaking, we do something like this. We have an observed image, which is the true image times the bias field. And here you see how it would look like in practice. On the left, you have the MR image before bias field correction, and here after bias field correction. Here in the middle, you see the intensity histograms, and there you already see like all of those peaks, which are scanner noise. Oh, sorry. And the lower image shows you the segmentation, and especially here in the frontal area of the brain, which had more a shadow of the bias field. You see that the distinguishing between white matter and gray matter was quite difficult. And after the bias field correction, you see a more clean segmentation of the brain. Nice. With the corrected bias field, we can now eliminate non-brain tissue. Therefore, we just get rid of all the tissues around the brain, such as skull, fat, skin, etc., etc. Therefore, we find the boundary between brain and non-brain tissue using image contrasts, create a mask. Here in the brain tissue is E equals to 1, and all voxels which are non-brain tissue are 0. And then we can cut out our brain image. Perfect. Now we can finally start with our segmentation of different tissue types. I already talked about those two different signals, the T1 and the T2 signal. And those two different signals, of course, also transform to two different MR images. Now, as an example, I talk about CSF. CSF has a relatively high water, so therefore the T1 time is quite long, and we have a low intensity in the T1 weighted image. On the other side, the T2 time would be quite short, and therefore we have a high intensity. If we now plot the intensity histograms next to each other, so like the T1, for example, at the x-axis and the T2 on the y-axis, we can easily differentiate the different tissue types. And here, for example, you see how you can, using a segmentation algorithm, segment CSF from gray matter, from white matter, and this is also visualized here on top. Just to mention some algorithms that normally are used to do this, like often you use hidden Markov random field models or mixture models added with some registration, just to name some buzzwords so that you can look them up if you need further information. If we now have the different tissue types, we can now register them to MNI space. As David already mentioned before, we have like those different coordinate systems where we somehow need to jump in between. And this we do by transformation. Here just a simple example. If this on top is our original image and we want to transform it to the lower one, we jump in X direction, we have a translation in Y direction, and we have rotation. all of this we can save in a matrix. Wonderful. Here we have rigid transformation. We can also add affine transformation, like sharing, like we add sharing. So we have affine transformation. This is still some linear transformation, so we can still all save it in a matrix form, which is wonderful to do follow-up analysis. However, we also need nonlinear transformation for image processing. And there we change the proportions of different objects inside of the image. Now talking about MR imaging precisely, if we look at our brain scan in the beginning, we first use affine registration to align the outer parts of the brain. Now the brain is still unhappy because the inner parts are not perfectly aligned, and therefore we use nonlinear transformation. And tada, we have a happy brain. All brains are in the same space, and we can compare them with each other. In real life, this would look like something like that. Okay. And our last step, finally, the surface reconstruction, then so that we can afterwards parcellate our brain. Well, the problem now is that we have been doing all of our computations in a three-dimensional space, right? So we were always talking about volumes, about voxels. But now we somehow need to come from this volume space or from these voxels to a surface. More precisely, we want to know where is this blue surface, the white matter surface, and where is this red surface, the surface between gray matter and CSF. And this we can model with a 2D triangular mesh. Why triangular mesh? Well, triangles have an extremely simple geometry and they are always planar. What I mean by that, if you draw 3 points in a three-dimensional space, you will be always able to draw also a 2D surface through them. If you now try to randomly put 4 dots in a 3D space, it will be highly complex to draw a surface between all of them. Additionally, you will be also able to construct more complex 3D objects with triangular meshes, which honestly, with all those foldings, a brain surface is as well. For this, normally you use tissue, you convert the tissue boundaries into, like the tissue boundaries here that we see here, into those triangular meshes. And so you have at the end the wonderful surface you can use. Okay, this surface that we reconstructed now, this is the upper image. This one is the red boundary, and this one would be the blue boundary. We use now the pile surface inflated so that we don't have all those tiny folds of the gray matter, because then we can align it with our parcellations, our atlases. Here, for example, you see the Desikan-Kiliani atlas. That's the same atlas that Luca already used in his experiments that he presented to you on Thursday about neuroplasticity. And this you can afterwards convert back to our subject space or unfold it in MNI space, and then we have our wonderfully parcellated brain. And with this result, here we see again, David talked in the beginning about different file types, right? And here you see again, to close the loop, why we need them. This surface, for example, is saved as SIFTY, and these volume spaces are saved as NIFTY. And we will look at this again a bit more in detail during our hands-on session. Okay, now we have our wonderfully parcellated brain. What do we need? What do we need this for? How do we get into TVB? Well, this parcellation itself will not immediately go into our model. We need this parcellation to feed it into the other two pipelines. Basically, we now have a map on which voxels of our fMRI data will then belong to which parcel of our brain, which afterwards we group to one single node in our network model. This said, we can now go further to the functional MRI processing pipeline that David will talk about.
Yes. Okay. Next slide, please. Okay, so when we analyze fMRI data, most of the statistical techniques we use rely on certain assumptions about the data. And this is mainly that the whole brain is sampled at the same time, that the signal intensity of a given voxel refers to the same location in the brain throughout all the volumes in the fMRI time series, and that the intensity of the fMRI signal we record reflects real changes in blood oxygenation as a proxy for neuroactivity and not magnetic or other artifacts. And with the raw data from the scanner, none of the assumptions are true, and the goal of reprocessing is to satisfy them. So, next slide. I will talk about the number of steps which are part of almost all fMRI preprocessing pipelines, and the first one of those is slice timing correction. Next slide, please. Okay, so the goal of slice timing correction is to satisfy the assumption that the whole brain is sampled at the same time. And initially, this is not true, because we scan the brain one slice at a time, and so different slices of the same volume are required at slightly different time points within the repetition time window, as you can see in the image on the right. And since this could confuse our later analysis, we try to correct for this using temporal interpolation. And the way this works is that we pick a single reference time point within the repetition time window, for example, the beginning of the time window, as you see on the right. And then for each slice, we try to predict what the signal would be at this reference time point based on the actual recorded signals at the time point surrounding it. This gets us much closer to the assumption that the whole brain is recorded at the same time than the raw data itself. Next slide, please. The goal of motion correction is to satisfy the assumption that the same voxel refers to the same location within the brain throughout all the volumes. And this doesn't tend to be true since participants always move within the scanner. And even small movements can have large effects, because especially at tissue boundaries, there are large differences in the signal intensity of neighboring voxels. So even a movement equivalent to the size of a single voxel can lead to up to 5-fold increase or decrease in intensity. And these movement-induced changes can then lead to spurious results in the statistical analysis. For example, the typical pattern of false activation at brain boundary you can see on the graph. Next slide, please. To correct for these effects, we usually choose one reference volume within the time series and then align all the others to it. This alignment is done by calculating which rigid body transformations that Luise already mentioned will minimize the difference between each volume and the reference volume. Next slide, please. Once we calculated these transformations, we apply them and register all the volumes to the reference volume. The problem is that the voxels before motion correction correspond to different locations within the brain than the reference volume voxels. So when we rotate the image, or when we transform the image using the rigid body transformations, it has the right position and orientation, but we know the signal intensity for slightly different points in space than in the reference image. This is not good for further analysis, and therefore, before we proceed, we need to use spatial interpolation to predict what the signal intensity would have been in the locations corresponding to the reference volume voxels. Next slide, please. So this alignment procedure for motion correction is not perfect, and some motion artifacts remain even after it. But by calculating these motion parameters, these rigid body transformations, which you can see on the right, we can save these and then later use them during denoising to regress out the residual motion effects from the time series. And we will talk more about this during the hands-on session. Okay, so the next processing step is susceptibility distortion correction. And the goal of this one is to remove magnetic artifacts. As Luise already mentioned, the magnetic field within the scanner is not homogeneous, and the differences in field intensity can cause distortions in the fMRI images, which you can see on the left. On the front of the brain, you can see warping either inside or sort of outside. And in the center, you can see the picture of an inhomogeneous magnetic field that causes the distortions. And an image like this that captures the intensity of the magnetic field is called a field map and can be produced during image acquisition and later used to correct for the distortions, as you can see in the corrected brain on the right. Next slide, please. So the previous three steps satisfied some of the main assumptions necessary for successful statistical analysis. However, there are still some problems that need to be solved before we can proceed. And one of them is that fMRI shows us changes in activity, but it has low anatomical detail. And therefore, if we want to localize where the recorded changes in activity are happening within the brain, we first need to map the functional image onto the structure image. This is called co-registration. Next slide, please. And then even once we have co-registered functional and structural images, we still face the problem of different brains having different sizes and shapes. So we need to normalize the image we have. Next slide, please. Okay, so these pre-processing steps I have talked about are part of most pipelines. And once such pre-processing is done, the subsequent steps depend on what sort of analysis we want to do. For TVB, we are usually interested in the functional connectivity analysis. And there, the most common pre-processing steps are, or post-processing steps, are denoising, where we try to remove different sources of noise from the time series. And we'll talk about this in the hands-on session. Then smoothing, and finally, the parcellation, using the parcellation to calculate the average time series of each region of interest. Next slide, please. Once we have this average time series for each region, we can calculate correlations between them. And those correlations are the functional connectivity values which form the FC matrix we will later use in TVB. Okay, next slide. So we hope that now we have a good picture of how you get from raw data to the ingredients that you need for modeling using TVB. And I know it might seem like quite a lot and quite overwhelming, all these details we told you. And MRI processing is indeed quite a complex field and basically a research field of its own. And we are not telling you about this because you need to spend years learning about this, but so that you have a nice overview of the processing steps which maybe happen to data you are using in your research, so that when you run into issues, you maybe know what questions to ask, what to consider. Yes, and that's the end of the theory part. And so if you have any questions, go ahead.